go ahead and get started this Thursday afternoon. A few quick announcements. Homework 1, many of you, over 75% of you, turned it in for extra credit. That's awesome. Keep that up. Homework is only extra credit. If you never turn it in, it will not hurt your grade. But if you do turn it in, it can really help your grade, and that can make a difference in the end. So really strive to get it in and at least get a half point by turning it in by the following Tuesday at noon. At that time, I then post the answer key, and that's why we can't accept any more for credit once the answer key has been posted. If you missed last week, no big deal. You'll have another opportunity with the second homework being posted right after class today. It's probably a good idea every time you submit something on Canvas for any class to check what you have submitted and make sure it is what you intended to upload and submit. If there was a mistake or a problem with what actually got uploaded, you can always send an email to the IAs or me and can fix the problem. About 30 of you have tried out the practice online Canvas exam, so that's great. You know it's working for you. The other almost 200 of you, I highly recommend you do this well in advance of the pre-midterm. I know that's not for another 2 weeks, but if your computer or your site is glitching and things are not working, it could take a week for IT to figure that out for you. So just try it out. It's anonymous. You don't get a score, or you get a score for yourself, but I don't see it. But make sure everything is working before that pre-midterm opens. And then finally, I've added another module onto the Canvas site. So when different groups send me information that might be of use to some of you, I'll be depositing there. And so check it out on a regular basis. Any questions on class format or these issues? All right, so there we go. All right, so class on Tuesday, we spent the bulk of it talking about this first major molecular mechanism that we'll learn in the class, and that is DNA replication. DNA replication is just DNA being copied into more DNA. And we went through this complicated figure showing how DNA is replicated and talked about all the various factors and their activities that are involved in copying the DNA. The next few slides I'm going to go through sort of breaks this down and simplifies it even further. Those slides are not currently in your notes because I really just want you to think and pay attention to what I'm showing you. But every slide that I'm going to show now that's not in your notes will be in the clicker notes that I post tonight. So you will have full access to that. All right, so for DNA replication to occur, the first thing we're going to need is a sequence or an element in the DNA that tells the factors to come in and start the process. We call this element or sequence an origin of replication. What is the factor that's going to bind to the origin of replication and start the whole process? Helicase, exactly. We need to unwind the DNA, break those base pairs so that we'll have the single strands that will serve as the templates for new DNA synthesis. And as helicase is unwinding that helix, the double-stranded DNA ahead of it is going to be under some stress, and so there's an activity, an enzyme called Topoisomerase that will relieve that stress. Now, when we have unwound our DNA, we want to use both single strands in order to copy and do synthesis in the 5' to 3' direction. What that means is on one half, we're going to do leading strand synthesis, can just be continuous synthesis. And then the other half is going to have to go through discontinuous or lagging strand synthesis in order for synthesis to be in the 5' to 3' direction. So bottom strand here, is this going to be undergoing leading strand or lagging strand synthesis? Leading strand, exactly. And so this side will be lagging strand. All right, so we want to start synthesis. We have unwound our DNA. So those bases, the hydrogen bonds have been broken, but they want to snap back. They want to pair back to each other. So what is the factor that's going to need to bind to make sure that doesn't happen? Heard it. So it's called RPA, or replication protein A. But if you just remember it is a single-stranded DNA binding protein, that's fine too. It's called RPA in human cells. It might be called something else in other cell types. But the main point is to understand that once you have unwound or denatured that DNA, you need to keep it apart long enough to be used as a template. So RPA is going to coat that single strand so it can't snap back together. And then what do we need to happen? So we're ready to start synthesizing, but what is the first thing we need to make? A primer. And what is that primer made of? It's RNA. Weird. We're doing DNA replication, right? But actually we're going to make an RNA primer. And what is the enzyme that makes that RNA primer? Primase. Primase is a type of RNA polymerase. RNA polymerases don't need a primer. They can just start taking those nucleotides, base pairing it to the template, and forming phosphodiester bonds. So after Primase lays down an RNA primer of 15, 20 nucleotides long, then what takes over synthesis? DNA polymerase hops on, and since it's the first DNA polymerase that works on either leading or lagging strand, which one is it? Alpha, right, beginning of the Greek alphabet, alpha. So on both leading and lagging strand, we're going to need to lay down a primer. Primase puts down the RNA part, and then DNA polymerase, alpha, will extend that by about another 20 nucleotides long. All right, so if we are on this strand, which we already said is going to be leading strand synthesis, once we get this initial primer laid down, what's going to take over next? Epsilon, DNA polymerase epsilon, and it needs some help to get clamped onto that template. Does anyone remember the names of those factors that help clamp it onto the template? RFC, replication factor C, and then PCNA was the name of the other one. And so that will clamp epsilon on. And then up here, the same thing's going to happen with those polymerase loaders, except for which polymerase is going to work on lagging strand? Delta, exactly. So leading strand synthesis, we have just extended this primer, and we have polymerase epsilon, and it is just going to follow the helicase as it unwinds and reveals more template. On the other hand, we have to keep laying down RNA primers followed by polymerase alpha in order to have 5' to 3' synthesis on the lagging strand. Once that primer is laid down, DNA polymerase delta will extend it until, it's going to run into, we have RNA still in our DNA. We need to get rid of that initial primer. What is the name of the factors that get rid of the RNA? RNase H. So RNase means it's going to degrade RNA. H actually stands for hybrid because it's a hybrid between the RNA primer and the DNA template. It will degrade the RNA part of that. Fen1 is also involved in this, and actually the entire primer that was put down by Primase and alpha tends to be removed. So these enzymes come in, they remove the primer. That also would have happened in the leading strand as well. Once that is removed, DNA polymerase delta up here in lagging strand can then use this exposed template to continue synthesis until it reaches the DNA that had been laid down in front of it. And so once it copies all the way, puts in the last nucleotide that's complementary to the template, needs to make that final phosphodiester bond. What is the name of the enzyme that does that? Ligase, exactly. So ligase will seal that. What about down here? Well, if you remember, when you have these replication bubbles, you're going to have leading strand, but eventually lagging strand from the other direction will catch up, and then that is how that gap would be filled. Does that make it clear how this process works? And like I said, all these slides will be in with your clicker slides tonight. Any questions about DNA replication? All right, let's do a clicker question. So I'm just showing you a single-strand template sequence up here and asking you, which of the following could serve as a primer for replication of the above strand of DNA? Ten more seconds. All right, final answers. All right. We have a lot of people liking A, not many votes for the middle ones, and then also a fair amount liking E. All right, let's figure out what is the correct answer here. Who wants to share their reasoning and answer for this? Everyone's feeling a little unsure. There's a lot of votes for A, but I kind of liked E, or E seemed good, but maybe it's A. Maybe it's one of the middle ones. Go for it. Right, so we want to make sure that our primer is going to base pair anti-parallel, so 5' to 3'. And so she went to the end here and said, okay, this 5' AUAA will base pair here, and it'll base pair all the way through. And then the 3' will be here, and then I can copy the template. That is the correct answer. Many of you chose this answer. And yes, it is true that this will base pair here, but are you going to be able to replicate or copy this template if the primer is base paired that way? No, because synthesis is always 5' to 3'. You're always going to be adding onto this 3' hydroxyl. There's no way to copy that DNA if you base pair at that position. And that is why E is the answer, because it is the only one that will base pair in the correct orientation here. Here's your 3' hydroxyl, and then you can just copy the DNA template in this direction. Does that make sense to people now? So when we want to replicate DNA, that means we want to copy the DNA. And so therefore, we always need to be adding on to that 3' hydroxyl using the template sequence. Any questions? All right, so then at the end of class on Tuesday, we started to talk about this end replication problem that happens at the ends of our chromosomes, where we will lose sequence through each round of replication, and we need to have these stable structures at the end. And we call these stable sequences, these repeat sequences, telomeres. These are these sequences at the ends of our chromosomes. And the way that they are synthesized and maintained is through a special enzyme called telomerase. Telomerase has two subunits. One of it is a protein, and one of it is an RNA. What is the role of the RNA in the telomerase enzyme? What does it serve as? Yes? Yeah, exactly. It is the template. It has that sequence that will be copied and repeated over and over again. The template is always what gets copied. So yes, the role of the RNA is to serve as a template. What about the protein? What kind of protein or what kind of enzyme is it? It is a reverse transcriptase, because we just said we're using an RNA template to synthesize DNA. The DNA that's being synthesized is telomere sequence, right? We are just adding on to the 3' end of this single-stranded overhang at the end of our chromosome. So the telomerase enzyme, using a special portion of its RNA, will come in and base pair to that repeat sequence and act as a template. So right here, there's a 3' hydroxyl. And so the telomerase enzyme will then be able to add a T across from this A, T, C, moving in this direction. Once that template region has already been all filled, then the whole complex slides down the primer, right? The telomere is serving as a primer. It has that 3' hydroxyl. We call this translocation, and now there's more sequence of template available. You'll keep adding on to the 3' end, and this can just go on and on and on and make a long single-stranded repeat sequence. And then eventually your normal lagging strand DNA replication machinery can see this template sequence, lay down a primer by primase, alpha takes over, delta hops on, synthesizes, that last phosphodiester bond will be made by ligase. Any questions about the general mechanism of telomerase? Yes, Lydia. Yeah, so the RNA itself, the RNA component of telomerase, is usually hundreds of nucleotides long, but only a small segment, what is in bold here, only that is used as the template. It's the only part of the enzyme that is accessible and can be read as a template and just keeps sliding along. And that's why we get the repeat sequence, because the same little piece of template is used over and over and over again. Other questions? All right, so the other way that telomeres are protective is they don't only just put in all this buffer repeat sequence, but the very end, the parts that's always going to be single-stranded can actually induce this sort of looping structure at the end of the chromosome, base pairing to another part of the telomere sequence. And then there are a whole bunch of proteins that recognize this repeat sequence and bind to that. Don't worry about the names of the proteins. This general complex is called the Shelterin complex. Shelter means to protect, right? And so when we're thinking about how telomeres protect the ends of our chromosomes, it's not just by adding all that repeat sequence, but also because there is a specific DNA structure and proteins bound to it that really help protect those ends so they don't just look like loose linear pieces of DNA. All right, so we've talked about this. Our telomeres are important. They protect the ends of our chromosomes. If you just had a linear sequence there, that would be very bad. It would look like a piece of broken DNA, which we'll talk about in our DNA repair mechanisms later in class today. So they're really important to have your telomeres, and therefore telomerase must be really important and active and make sure we keep our telomeres, right? What do you think the cellular expression of telomerase is in your cells, your adult differentiated cells right now? You have really high levels of telomerase in there working hard to keep those telomeres? No, actually telomerase is pretty low or not even active in the vast majority of your adult differentiated cells. Only cells that need to rapidly divide, that are replicating a lot, like your stem cells, your bone marrow, your germ cells, they have high levels of telomerase. The rest of your cell types pretty much have silenced telomerase and won't have it on. And so this was kind of surprising when people first noticed this, but it's like, all right, well, if a cell's not dividing, I don't have the end replication problem, so, maybe that's okay. And this actually solved a mystery that was identified now over 60, 70 years ago, when scientists were able to take cells, so scrape some of your skin cells, put it in a culture dish, give it all the nice nutrients that they might want, and those cells will divide. Each division involves DNA replication, but only for about 50 divisions, and then they would stop. They would senesce. They would go to sleep. This is sometimes called the Hayflick limit. Some of you may have heard of that. That's because Hayflick was a professor who noticed this. And the reason is because those cells don't have telomerase active. And therefore, with each round of replication, their chromosomes are getting shorter. They go into a crisis, and then they stop dividing. What was realized is if you just then express telomerase in these culture cells, they will divide forever. So almost all the culture cells that we use in the laboratory have telomerase active so that the telomeres can be maintained on these cells that are being used, that are being replicated in the laboratory. All right, so it's kind of making those cells immortal, right? They're living forever. So maybe we should turn telomerase back on in all of our cells, make us live forever, right? Because even though our cells aren't dividing as we age and get older, those ends do start to whittle away. Our telomeres do start to get shorter. So we should buy this telomerase activator that you can buy, right? And make sure that we have really high telomerase levels everywhere. Is that a good idea? I'm hearing no. Why? So it turns out that 80 to 90% of all cancer, so cancer is tumors, right? And a tumor is just cells dividing out of control. So a whole lot of DNA replication needs to go on, need to keep dividing. Tumor cells need to maintain their telomeres. And so something like 80 to 90% of all tumor cells have found a way to reactivate telomerase, or have they found another way to maintain their telomeres. So instead of taking these supplements to become immortal, scientists started to think, aha, well, maybe this is a magic bullet. Maybe if we inhibit telomerase, tumor cells will die and not be able to replicate. And there was a lot of investment and a lot of companies that have tried to do this. It has been largely unsuccessful, unfortunately, except for recently there's one type of blood cancer where there has been efficacy in inhibiting telomerase in that particular cancer and inhibiting the telomere formation in that cell type, and it seems to be working for some of these patients. So maybe in specific types, hitting the telomeres and hitting telomerase might be useful, but most definitely don't buy these supplements, okay? All right. Any questions about telomerase or telomeres before we move on to new material? Okay, so now that we've learned about the basics of DNA replication inside our cells, we're going to learn how we can use that knowledge to replicate specific sequences of DNA in the laboratory. And this is a method called the polymerase chain reaction, or PCR. I think when I asked on the first day if anyone had done that before, many of you have done this already, so this may just be a review for a lot of you. But then we'll talk about, once we understand how to do PCR, how it can be used in DNA fingerprinting. And if you really want a nice animated version of PCR with good music to it, I recommend you check out this video here. I also posted one of these classic experiments that talks about discovery of PCR and the type of polymerase used for it on the class website. All right, so the goal of PCR is simply to amplify a specific DNA sequence. So if I isolated the genomic DNA from your cell, so the total DNA sample, and I just wanted to amplify the globin gene, I could do that through this PCR reaction. So thinking about how we did replication within our cells, what do you think are the requirements? What do I need to put into my tube in order to do a PCR reaction? If I want to copy a specific sequence of DNA, what am I going to need? Yes? You're going to need primers. Whenever we're doing DNA copying, we're going to need some primers, and we'll talk about those types of primers in a moment. Yes? Template DNA, yes. And in this case, it could be the entire sample of genomic DNA from within a cell. So we have our template, we have our primer. What else do we need? Yes. We need a protein. Yeah, we need protein to do the work, to do the synthesis. And what's it going to use to do the synthesis? Yeah, what kind of nucleotides? deoxynucleotide triphosphates, or DNTPs. So we need some kind of template telling us the sequence that needs to be copied. We need primers. When we do PCR in the laboratory, we use DNA primers. These can just be chemically synthesized. We actually buy them from companies. Usually they're at least 20 nucleotides long. So by oligo, I mean a single strand of DNA that's serving as a primer. We need deoxynucleotide triphosphates. And then we need a polymerase to do the work. And we have a special polymerase that we use in the PCR reaction. That's what I mean by this asterisk here. Because as you'll see in the next slide when we talk about the cycles, this polymerase has to go through some pretty wild temperature changes. And most proteins aren't very stable under high temperature changes. But the polymerase that's used for PCR actually is. And so the 3 steps that go into each cycle of PCR are shown here. So we said we need our template DNA. It's going to initially be double-stranded. In order to copy DNA, we're going to need to make it single-stranded, right? So we need to denature that double-stranded DNA. And how do we denature that DNA in a PCR reaction? High heat. We crank it up to almost boiling, okay? So this is why we need a very stable polymerase. So that will denature the double-stranded DNA. We then bring the temperature down to a level where primers can then base pair. So these are these DNA oligonucleotides that we have designed and ordered from a company. They are in excess, meaning there's a lot more of the primers than there is of template, and that's why they are able to then base pair to the template before the template DNA comes back together. All right, we have our primers base paired to the region we want to amplify. And then the last step, this is where that polymerase, TAC, thermus aquatis polymerase is a common one. It's very stable at the high temperatures. It will come in, see that 3' hydroxyl, see that 3' hydroxyl, and add the deoxynucleotide triphosphates, moving in this direction and moving in this direction. That's how we get our new, we've gone from 1 double-stranded piece of DNA to now 2 double-stranded pieces of DNA. All right, let's make sure we understand how to design primers in order to do this type of PCR reaction. So let's say the sequence happens to be at this position and this position where we want to start designing our primers. So after we've denatured and annealed our primers, what do we want the sequence to be of this dark blue primer? What would the first 3 nucleotides be? And what would you write first? Oh, it's already written. 5', A, T, G, right? You want it to base pair to the template. And lo and behold, it's exactly the same sequence that it was originally paired to, right? It's just using the base pairing rules. What about on this side? We would have 5', C, T, A. Exactly. So you just use your base pairing rules. The thing that's really important to keep in mind is the 3' hydroxyl needs to be facing in the direction you want synthesis to happen. So always, always write your 5' and 3' when you have to design a primer sequence, and then just make it complementary to the template. Lydia. Let's get to that question. So I said in the last slide we're going to use DNA primers that we just bought from a company. But in a few slides I'm going to ask you why we don't use RNA primers like our cells use. So hold that thought for a moment. All right, so our first round, we would have our primers here with those sequences, and then look and see what happens. So once this 3' hydroxyl gets added onto and the polymerase copies, what's the sequence it's going to copy here? C, A, T, which is exactly what was in the original strand, right? So what we have done is we have made one of these into now 2 of these. And then what's beautiful in the PCR reaction is that is going to be 1 cycle or 1 round of PCR. What's going to happen next? You denature. You just go back, and now all of this double-stranded RNA is going to get, or DNA is going to get denatured. You have lots of primers still in your test tube. They are going to base pair. Your polymerase is going to add DNTPs in that direction. And the process is going to go over and over and over again. And if you repeat it 20 times, that's about a 1,000,000-fold amplification. This is how we amplify a specific sequence of DNA. And what's nice here, and the reason why we get this amplification, is because after each round, we have essentially made more template for the next round. Each newly synthesized strand of DNA will be a template for the next round of synthesis. And if you keep doing this over and over and over again, what you see here is the vast majority of your products are going to be exactly the size based on how you designed your primers. Where your primers sit, that's going to be the size you get. Because as you can see here, when you get synthesis, for example, if this primer used this template here, base paired here, once it gets to the end, where that primer originally was, there's no more sequence to copy. So after many rounds, this is the vast majority of the product that you will get. Does everyone see that? All right, now let's get back to Lydia's question. So why are DNA instead of RNA primers used in our PCR reaction in the laboratory? Sure.
One of the reasons is DNA needs a way higher temperature than RNA. So like the 95 degrees, like if it was at RNA, it would probably like denature a lot less. The other one's at 72.
So actually, RNA primers would anneal just fine at these temperatures. So they would anneal. If this was RNA, it would anneal. But what would be a problem? What is our goal of this PCR reaction? Why are we doing PCR? To amplify a specific sequence of DNA, right? What happens to the primers? They become part of your product, right? Yeah. So we don't want RNA if we're trying to make DNA, but there's an even bigger problem. So you are copying the sequence of the primer when you have synthesis going in this direction. If this were RNA, could Taq DNA polymerase copy RNA? No, it is not a reverse transcriptase. It is a DNA polymerase. It uses a DNA template to synthesize DNA. So we do not use RNA primers when we do PCR in the laboratory. One, because we want our product to be all DNA in the end. We don't have RNase H in here to take that out and like get filled in with something else. And then also, our Taq polymerase wouldn't be able to use that primer as part of a template. Does that make sense? Questions? All right, so this is just a review. So the whole goal of PCR is to amplify a genomic region of DNA. So, if I collected all the DNA from your cells, billions of base pairs of DNA, and just wanted to amplify your globin gene, maybe to sequence it, see if there's any mutations in it, I would just design primers for that specific region, go through the multiple rounds of PCR, and now I have many genomic clones of that specific sequence. And so PCR is, of course, used in forensics. You've probably heard in your television programs, they're always going to do a PCR test to identify or rule out suspects. And so this is in a method that's often referred to as DNA fingerprinting. Just like we all pretty much have unique fingerprints, we also have a unique pattern of DNA banding. And it is going to be based on our simple sequence repeat regions. Remember, we have all these repeats throughout our genomes. We have those small areas of repeats that can be like AT, AT, AT over and over again. And so these sequences of the simple sequence repeats can be highly conserved. So like the example of AT, that sequence, AT, AT, that sequence is conserved. But the number of repeats among individuals can differ. And PCR is a great way to identify whether that size of the repeat region is different. So, for example, we have genomes from 2 different samples here. This 8-unit array, you can imagine this is AT repeated 8 times, so AT, 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 versus in this sample, it is AT repeated just 4 times. If I do PCR, I hope you can imagine if I design primers on either side of the repeat region, this is going to be a bigger size PCR product than this one. Is that clear? And now that we have the whole human genome sequence, we know that there's over 20,000 different simple sequence repeat loci throughout the human genome. Typically, when we do DNA fingerprinting to identify individuals, we do 15 to 20 specific core regions. And if you do the math and do that many, that's actually the odds of that being the same between 2 people is over 1 in 1 trillion. Right? There's not a trillion people even in the world. So it's very specific. Here's an example. So here you have 2 chromosomes. Maybe one's from the mother, one's from the father. Here's the repeat region. There's more repeats than there are here. So if you design primers on either side of the repeats, you're going to get different size PCR products. Is this chromosome the paternal or the maternal based on how it has run in this electrophoresis gel? It's paternal, right? Because it's bigger than this one, and bigger things migrate more slowly through a gel. So therefore, this bigger band must have come from this one. So that would be the paternal region. When you are designing your primers to do this type of DNA fingerprinting, it is very important to make sure that they are flanking or on either side of that repeat region. Why don't you design your primers to be in the repeat region? Because they could base pair here, 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 right? It's a repeat region. When you design your primers, you want them to base pair to just one separate place within the genome. You design them to be very specific so you can predict exactly what size or what region that you are amplifying. Any questions about that? All right, let's look at a more complicated examples, because as I mentioned, typically when this DNA fingerprinting is done, they use multiple sets of primers. So they're looking at maybe 15 different simple sequence repeat loci in one reaction, right? So in that reaction, you just have the DNA sample, and then you have multiple sets of primers that will base pair in various regions of the genome, your deoxynucleotide triphosphates, and Taq polymerase, and you'll do the PCR. So here's an example of a paternity test, and so I'm asking you here, which sample is from the child's father? So this is the mother's fingerprint, DNA fingerprint. This is the child's. Here's father 1. Here is father 2. So which one do you think is the father in this paternity test, and why? Why F1?
If it doesn't match the mother, it must match the father. And then you do that if the children doesn't match it.
Yeah, exactly. So the child, of course, is going to be a combination of mother and father. So a band from the child should match either the mother. This one doesn't match the mother, but it matches father 1. Kind of doesn't match father 2 there. Here we have child matching mother. Here we have child matching father 1, child matching father 1, and so forth. So every band in the child matches the mother or one of these fathers, and we can definitely rule out father 2 here because it doesn't match actually at any of the positions. Question?
Is it conclusive?
Yeah, with the number of bands that they look at, it's, yeah. I mean, nothing is 100%, but I would, 99.9999999% believe a paternity test based on DNA fingerprinting.
Is it like number of bands that must match? Like minimum number of bands?
So, I mean, if you have ones that don't match and don't match also the mother, then like you can definitely rule out. You can rule out definitely. Put it that way. All right, so DNA fingerprinting isn't just used in paternity tests, but it also can be used in crime scenes. So here's a clicker question with, again, some DNA fingerprinting results, this time looking at results from the victim themselves, a specimen taken at the scene, maybe some blood sample, and then 3 different suspects. So which of the following can be concluded from this DNA fingerprint analysis? All right, put your final answers in. Ready? All right, so we have a lot of takers for E. It's got to be more than one of these above, but A seems to be varied, and some votes for the rest of these. So let's see. Okay. Who wants to explain their reasoning for this? Sure.
I don't know if it's where I'm sitting in the corner, but
Oh, yeah.
it kind of looks like for suspect 1 and 2, a lot of the stuff does line up with the specimen. I don't know if it's like because, you know, it's at an angle I'm seeing it, but I definitely can see that. I can see that with the victim, the specimen. It does look like there are some points that line up. Again, I think it's where I'm sitting, but kind of based off of that, that's the best of my knowledge. I can say there's 2 of them.
Okay, all right. So you brought up some important points here. So let's think about this. So the specimen. So this is different from the paternity test, right? So if you leave your DNA at a crime scene, should every band in that specimen also be found in your sample? If I leave my blood somewhere and someone does DNA fingerprinting, every band from me should be there. Could there be additional bands? Yes, maybe if they did a DNA fingerprint of this whole class, it'd be very complicated, right? We'd have so many different bands, right? So with the child in the paternity test, we had the child only half of the bands would be mother, half of the father. So it would not match anyone in particular. So this is a little bit different, right? So with that in mind, let's go through each of these options. So we had a lot of interest in A. A says the DNA from suspect 3 is not found in the specimen from the crime scene. So here we have suspect 3. Are all of their bands found in the specimen? No. There's a few, but not all of them. So that rules out that this suspect left the DNA that is in this specimen. They still might have been at the crime scene, but their DNA is not contributing to this specimen profile. Does that make sense? All right, let's keep going. B says DNA from the victim is represented in the specimen. So same idea. Here we have victim. Is every one of their bands also found in the specimen? No, there's several that are not found at all, so the specimen is not contaminated with victim DNA in this particular case. All right, what about C? C says suspects 1 and 2 left DNA in the specimen found at the crime scene. So same idea, we're going to look at suspect 1. Suspect 1 looking pretty good here for contributing to this specimen. Yeah, so every single band we see here, we see here. So definitely contributed to the DNA that we are fingerprinting in the specimen. What about suspect 2? No. Suspect 2 has bands that are not found in the specimen, so suspect 2 is not contributing to that at all. So it looks like we have a pretty clean sample of suspect 1 right here. But since I said suspect 1 and 2, C is not correct. What about D? Multiple primer sets were used for the PCR in this DNA fingerprint analysis. Yes, absolutely. That's how I got multiple bands, right? So you have a set for this simple sequence repeat on this chromosome, another set for another simple sequence repeat, another set here, and that's how we get these banding patterns. So the ultimate answer was more than one of the above. It was D, and it was A. Does everybody understand that?
What does D mean? Multiple primer sets were found.
So what is a primer? So what are primers? That's how we amplify. We start the DNA synthesis, right? Those need to base pair to the template, so we get synthesis. So one set would amplify one position. A second set would amplify another position. A third set would amplify another position, right? So we have many bands here, so we use many sets, maybe 20 different sets of primers. Yes, yes. Question? So if you leave your DNA behind, you choose to just leave a few chromosomes? No, your entire sample will be there. And so therefore it should also, these bands would have to be there. Any bands that you see in suspect 2 would also have to be in the specimen for them to have contributed to this DNA fingerprint pattern. Skin, blood, hair, semen, spit, you're leaving your DNA everywhere you go. And we have more and more sensitive methods to detect it these days. So you leave that behind. Any other questions on this? All right, so some of you might have been thinking back, oh, we have this new PCR method that we now can do, and maybe I could actually apply that to my DNase protection assay. So remember, we talked about the DNase protection assay is a method to simply ask, is a region of DNA protected and therefore protected from DNase and compact, or is it a more open, loose, and therefore going to get chewed up by DNase? And in our DNase protection assay, when we talked about it, at the end we needed to be able to detect our sequence of interest. We were looking at the globin gene. We would design a probe in order to detect whether there is still globin gene DNA there at the end of the southern blot or not. But could we also use PCR to ask the same question? So here's what we learned in class, in the previous class using a southern blot. Could we just design primers across the globin gene? If this was not protected, what would happen to the globin DNA? Gets degraded by DNase. Would there be any template for the primers to amplify? No. You wouldn't get a PCR product, or you would get a very faint PCR product. If it was very, very protected, would you have a lot of template? Would you get a lot of PCR product? Yes. So many times we use PCR if there's just a specific gene we want to ask, was it protected in our DNase protection experiment? Does that make sense? All right, so now let's move on and talk about DNA repair, right? So it's very, very important for our DNA sequence to maintain the correct sequence, to pass this on. And so there's a lot of different mechanisms that your cells employ to make sure that changes to the DNA sequence, which could be mutations, which could be very dangerous for your health and for the cell. And so we'll talk about two general types of repair mechanisms. Well, first we'll talk about how our polymerases themselves can do some proofreading and fix any errors that they make. And then we'll talk about mechanisms that can fix a sequence if just one strand of the double strand is damaged, and then what will happen if we end up having a double-stranded DNA break. So the first thing is, well, what happens if the polymerase are, we have our high-fidelity polymerases, delta and epsilon. They're zipping along. They very rarely make errors. But one of the reasons why they're also considered high-fidelity is they can actually recognize when they make an error, when they incorporate the wrong nucleotide. So shouldn't have an A across from G, right? This should have been a C. But if this A gets incorporated, there's a chance that the polymerase will recognize this is incorrect base pairing. And what it will do is it will pause, go back, remove that nucleotide, okay? So since it's going back in this direction, we call it 3' to 5' exonuclease activity. It's going to remove that end nucleotide here. Once it removes that nucleotide, then it's going to resume replication, adding onto that 3' hydroxyl, hopefully putting in the right nucleotide. So synthesis, as always, will be 5' to 3' continuing along. And which are high-fidelity polymerases that have this activity? Delta and epsilon, right? They're good, but every now and then they miss their mistake. They're moving so fast and they miss a mistake. And so there needs to be another repair mechanism that is active during the process of DNA replication. And the repair mechanism is called mismatch excision repair. It finds those mismatches, the wrong nucleotide inserted, and is going to repair that. Don't worry about the names of the factors that are shown in these slides, but just know the general mechanism and as I've written it out over here. So the first challenge that this repair machinery is going to face is figuring out which is the wrong base, right? A is a normal base, C is a normal base. How does it know which was the newly synthesized nucleotide and therefore the incorrect inserted one by DNA polymerase? Does anyone know? Does anyone know how the new DNA is recognized? So old DNA, the template DNA, can actually have modifications happen to it over time. And so there can be also different proteins that are recognizing the old template strand. And so the repair machinery is able to recognize the newly synthesized DNA because it won't yet have these modifications or the same types of proteins bound to it. So the incorrect base on the newly synthesized strand is going to be recognized. And then the wrong nucleotide as well as surrounding nucleotides all get removed. And then finally, the gap is going to be repaired by your high-fidelity DNA polymerases. And then the last phosphodiester bond is going to be ligated by ligase. Question for you based on this mechanism. So during this mismatch repair, how does a DNA polymerase fill in the gap? And so the arrows indicate the directions of synthesis here. All right, put your last answers in. A strong battle between A and B. All right. Who wants to explain their answer? Yes. Yes. So which one? All right, so synthesis is always 5' to 3' like Madeline said. So is this going in the 5' to 3' direction, or is this going in the 5' to 3' direction? A. You are always adding on to the 3' hydroxyl. So find your 3' hydroxyl and then just synthesize in that direction. A is the correct answer, and it is synthesizing always 5' to 3'. Does everyone see that? Okay. Okay, so mismatch repair is how those misincorporated nucleotides get fixed during the process of replication. But most of our cells are not replicating. So are there mutations that can arise independently of DNA replication? Yeah, all the time. Our DNA turns out to be quite chemically reactive. In fact, there's some very simple chemical reactions that just involve water that can take, for example, cysteine, deaminate, remove this amine group, add this oxygen, and now we no longer have a cysteine, but does anyone recognize that base? It's uracil. Ooh, we don't want to have that in our DNA. That's a bad idea. This is, remember how I said DNA can have modifications happen to it over time? One very common modification is methylation at this position on cytosine. This is a normal nucleotide. It can behave normally. But when that cytosine, when that methyl cytosine gets deaminated, we no longer have something that looks like a cytosine. What is this? It's a thymine, right? So that would be definitely a nucleotide change in our DNA. So these simple reactions that just really involve water to catalyze them is happening to our DNA all the time. And if it didn't get fixed, we could end up with mutations getting fixed in our DNA. So in this example here, again, we might have our methylcytosine, which normally base paired to G, no problem. But then if this undergoes a deamination reaction, the methylated cytosine becomes thymine. If this is in an important gene, this could be transcribed into a messenger RNA with a different sequence that might make a protein that is different. Could be an oncogene, could lose a tumor suppressor activity. That's problematic. If that DNA gets replicated, then that change, that T, is now going to encode for an A when the DNA gets copied. So now you're going to end up with this mutant DNA sequence. All right. In this case, so T and G are both normal nucleotides. So how would the repair machinery choose which base to remove? Well, if you think back to the previous slide, I showed you that a simple deamination reaction can take methyl C to T. It's a one-step chemical reaction that uses water. What about G? So what base would G be most similar to? Is G a purine or pyrimidine? It's a purine. What's the other purine? A. So if you look back at those bases, G and A, there is no simple deamination reaction that would take G to A. It's much more complicated and doesn't happen to any appreciable level. So because this deamination reaction is so common, we have evolved enzymes to recognize when we see a GT mismatch in our double-stranded DNA, it is the T that is the mistake and needs to be removed. And it gets removed by a repair mechanism called base excision repair. And that is because it removes that base and just that single nucleotide and then puts in the correct one. So the first thing that happens is that nucleotide, in this case the T, is recognized as being the mismatch. That base gets removed. And then endonucleases remove the deoxyribose phosphate, so the rest of the nucleotide gets chopped out. You have a one nucleotide gap. There is a special DNA polymerase called DNA polymerase beta, beta for base excision repair. It puts a single base into that position, and then ligase seals that final phosphodiester bond. So that is for when we have simple changes in our DNA. However, there can be much more dramatic things that happen to our DNA. So which base is this? Guanine, right. So this is a G. If you smoke cigarettes or are exposed to a whole lot of cigarette smoke, there is this carcinogen that goes into your lungs, gets metabolized, and oh no, it covalently attaches to your guanines in your DNA. This is a very bad thing to happen, right? Because not only does it really distort your DNA, but how many base pairs should G make? 3. Can it still make those 3 base pairs? No, it actually gets read as a T, mutagenic. This is a huge problem. Here's another structural change that unfortunately probably happens to all of us every day living here in sunny San Diego. So what are these bases here? Thymine. So 2 thymines together, hit it with UV rays. Uh-oh, we get covalent bonds between these 2 bases. Does anyone know what this is called? When you have 2 thymers somehow stuck together now. So it's 2 and it's thymine, so it's a thymine-thymine dimer. You've probably heard of that before, so this is what your sunscreens are trying to protect you from. You don't want these to accumulate in your DNA either. Your cells really don't like this, and so we need mechanisms to fix distorted DNA. And the mechanism that fixes these distortions is called nucleotide excision repair. So what is nucleotide excision repair? Well, in this case, it's very easy to recognize what is the damaged DNA, what is the mistake that needs to be removed. For example, if you have these 2 thymines, these thymine-thymine dimers covalently ligated together, it's going to distort the DNA. This is recognized. There's a helix that comes in and opens that whole region of DNA. And then the distortion as well as the surrounding nucleotides are all removed. And then the gap is going to be repaired by your high-fidelity DNA polymerases, and then ligase will fill the final gap. Is synthesis going to go in this direction or in this direction? I guess that's hard to answer. The first one I said or second one I said? First, right? Here's our 3' hydroxyl. Polymerase is going to come in, use the template to know which nucleotides to add, and then the final phosphodiester bond will be sealed by ligase. So those are the 3 general repair mechanisms that we use if there is a mistake in just one side of our DNA, a single-stranded mistake. So we had mismatch repair when replication is going on, when the polymerase puts in a wrong nucleotide. We had base excision repair for those simple things that are happening like deamination events that are changing the base in the DNA. And then we had nucleotide excision repair when we have these distortions. Sometimes, though, we also have double-stranded breaks in our DNA, and this can happen from oxidative free radicals. This can be actually just products of metabolism. If you happen to be exposed to ionizing radiation, very bad thing, that can cause double-stranded breaks and mechanical stress. And sometimes we actually use cancer drugs to cause double-stranded breaks because some tumor cells are more susceptible to not being able to repair it, and it can kill the tumor cell. So because this can happen in our normal cells, we need to have repair mechanisms in order to deal with that. And we have 2 general repair mechanisms that will fix our DNA when we have a double-stranded break through it. One of them is homologous recombination, and the other one is called end joining. One of these is considered to be largely error-free, meaning that it fixes the mistake and there's no mutations left. The other one is error-prone and almost always leaves changes in the DNA. Which one is the more error-free one? It's homologous recombination. And the reason why homologous recombination is essentially error-free is it uses a homologous chromosome to serve as a template. Whenever we have a template, the polymerase then knows what sequence to put in. That's why our single-stranded repair mechanisms work so well. There's always a template there to know which nucleotide to put in the repaired sequence. You'll see where the homologous chromosome acts as a template in this air-free mechanism of double-stranded break repair. On the other hand, we have end-joining, which is an air-prone mechanism. And that, as you'll see, takes two broken strands of DNA, of chromosome, and ligates them back together. But that almost always involves the loss of nucleotides and sometimes even the addition of nucleotides. And even worse, if you have multiple chromosomes that have double-stranded breaks in the same cell, you can actually have two different chromosomes get ligated together. And this is called a translocation. And we can see an example of that in a few slides. All right, so let's look at homologous recombination first. So this is our air-free one because we're going to have a template. And the template for homologous recombination is simply going to be the undamaged homologous chromosome. So here we have, so in a diploid cell, you have two copies of each chromosome. And if you get a double-stranded break in one of those, then potentially the homologous chromosome can be used as a template. If you've just undergone replication and you still have your sister chromatids near each other, this is a very efficient process, homologous recombination. Like our other mechanisms, don't worry so much about memorizing the names of all these different factors. Instead, pay attention to the actual mechanism that I've summarized to the side of it. So it's pretty easy to recognize the double-stranded break. We have DNA that's broken in half. And then there are exonucleases that come in, and they actually chew away sequences, leaving 3' end single-stranded DNA overhangs. What do you think we're going to use a 3' end for? What can it act as? A primer, exactly. We're going to have a 3' hydroxyl. And there's a reason why it's been made single-stranded. And that reason is that single-stranded DNA end, so this single strand here, this dark green, what it does is it's going to search the homologous sequence in either the homologous chromosome or the sister chromatid for base pairing. It's going to come down and base pair the sequence in the homologous chromosome. Once it is base paired there, then the polymerase, one of your high-fidelity polymerases, can use that 3' end as a primer and add sequence on using the sister DNA as the template. And it's going to synthesize until it gets long enough, and then there's this complicated resolution. Don't worry about it. But what happens in the end is that you end up now with two intact pieces of DNA. We have now used the sister chromatid to repair this double-stranded DNA break, and it's now air-free because it had a template in the form either of the homologous chromosome or the sister chromatid if replication has just happened. So this is a repair mechanism we hope happens when we get double-stranded breaks in our DNA. But what are the odds of having your homologous chromosome close enough for this mechanism to efficiently happen in your cells that are not replicating? It's pretty low, so it doesn't happen that often, but you really got to fix a double-stranded break. And so to fix it in a hurry, we have this other repair mechanism, and this is called end joining. It essentially is going to put those two ends of the broken chromosome back together. So in this case, there's no template. The break is just recognized by the repair machinery. And during this repair mechanism, so as soon as DNA strands break, there's always going to be nucleases that are starting to chew away the DNA at that broken end. And so any overhang nucleotides that are left, those get also removed by the repair machinery until you finally get these blunt ends. And then they are ligated together by ligase. So as you can see, during this repair mechanism, you lose nucleotides. Strange things can happen in cells. Sometimes nucleotides actually get added onto these free DNA ends. So the result of end joining is almost always a mutation, almost always going to be a change in the DNA sequence that's going to involve loss of nucleotides, gain of nucleotides, combination of those two things. An even bigger problem can happen when you have multiple double-stranded breaks happening in a cell at the same time. What that means is there is the chance that parts of two chromosomes could actually get ligated together through this end joining mechanism. And we call that a translocation. And the example shown here is one that is commonly seen in a type of leukemia, where a segment of chromosome 22 has exchanged with chromosome 9. And so this is just scientists use probes to label chromosome 9 versus chromosome 22. And what you can see is there is this mix of chromosome 9 and 22 in this patient who has leukemia. And it was first discovered in Philadelphia, sometimes called the Philadelphia chromosome. Why is this a big problem? Well, it turns out that this particular translocation, where these broken ends of two different chromosomes have come together, actually make a new gene. It actually fuses this ABL gene. This ABL gene is a kinase that normally is very tightly regulated. However, in this translocation, it actually gets fused to this other protein called BCR. And when it is fused together, this kinase can no longer be regulated in the normal way. So now your white blood cells have this overactive kinase that say divide, 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 and then you get leukemia. So this is an example of a translocation which happens because end joining has put two different chromosomes together. And so these events can occur even in those of us who have normal repair machinery. If you expose yourself to too many situations that cause a lot of double-stranded breaks, you may overwhelm the system and end up with a translocation that can be quite dangerous. But there are also some poor individuals, some unfortunate individuals who are born with mutations that affect these repair mechanisms, and therefore they have heightened sensitivities. For example, some of you may have heard of this disease, xeroderma pigmentosa. These are these patients who can't be exposed to UV. They can't have any of their skin exposed to sun. And the reason is they have defects in nucleotide excision repair. Remember, this is the repair mechanism we need to fix distortions. A very common distortion in our DNA are those thymine-thymine dimers, which come when we're exposed to UV irradiation. These patients have a high susceptibility to developing skin cancer because they can't repair that type of mutation. BRCA1, many of you have heard of this. This is the breast cancer susceptibility gene. This is very important for repair of double-stranded DNA breaks by in cell types, in particular breast and ovarian. If you do not have effective homologous recombination to fix double-stranded breaks, all you can do is end joining. End joining is always going to be mutagenic. If it happens in a tumor suppressor gene or an oncogene, this can lead then to development of cancer in tumor cells. So you don't need to memorize these. I just want you to have an appreciation of how important these different types of repair mechanisms are, and when they don't work, what that can mean for human health. That's all we have for today. So do your homework, submit it for extra credit. Have a great weekend. Do the practice exam too. Make sure your computers work.